Welcome to Unit 1. Unit 1, we are covering the endocrine system. The endocrine system is the system responsible for producing hormones. Hormones are chemicals that are distributed throughout your body in order to mediate different activities that are occurring by different structures. And we're going to discuss how these chemicals are distributed and how they influence the activities in remote parts of the body. So let's first start off with a review of the different types of chemical signaling. The first is autocrine signaling. Autocrine signaling is when you have a single cell that secretes a chemical that activates a receptor and mediates that same cell's activity. So it's regulating itself, hence auto for autocrine. But then we could have a signal that's somewhere in between where it is influencing a neighboring cell. And this signal can jump through gap junctions. So a signal is produced here in the green cell and travels directly from its cytoplasm into the cytoplasm of a neighboring cell through a gap junction, therefore affecting the behavior of that adjacent cell. The next type of possible signaling is paracrine signaling. Now the cells are slightly farther apart. The signal is produced by the first cell and it diffuses extracellularly until it reaches the target cell that is nearby. It's within the same tissue. Finally, there's endocrine signaling. Endocrine signaling is the topic of this unit, where cells in a gland secrete a chemical signal that travels through the bloodstream and activates a very remote target cell, perhaps at the opposite end of the body. So there are lots of similarities yet differences between the nervous system and the endocrine system. The first similarity is they are both modes of communication, where one part of the body is able to communicate with a remote part of the body. For the nervous system, the communication is predominantly an electrical communication through action potentials down axons, but at the synapse, there's also a bit of a chemical signal through the release of a neurotransmitter. Endocrine signaling is only a chemical communication. There is no electrical component. And the chemical signal is released into the blood and moves to its remote destination. So because these are two very different types of modes of communication, they each have their strengths and their weaknesses and are optimized for different purposes. So, for example, let's talk about the speed and the persistence of a response. If a target tissue is responding to a nervous signal, where there was say, for example, an action potential in a motor neuron that then activated a muscle cell at a neuromuscular junction, the automatic response of that muscle cell is going to be quite prompt. It's going to start within 1 to 10 milliseconds. It's also going to extinguish fairly quickly. It's capable of shutting off very quickly. For an endocrine response, if you release a hormone into the blood, it takes a very long time for that hormone to diffuse throughout the circulatory system and reach the target tissues. And once it reaches that target tissue, the response ramps up slowly, but then will persist for an extended period of time. It could be weeks or even months. So there's a very slow onset and stop of any effects produced by an endocrine signal. Another contrast is how quickly the target tissue adapts to a long-term stimulus. If a nerve is persistently firing action potentials and signaling for a target tissue to respond, the target tissue will actually adapt pretty quickly and begin to ignore this persistent signal. An example of this is how quickly we adapt to sensory stimuli that are not changing. For example, the whirring of a fan in the room. If you were to hear a fan buzzing in the room, you would be aware of it the moment you walk into the room, but very quickly, after a few minutes, you become not aware anymore that the fan is there, that you've adapted to that stimulus. And there's a good evolutionary reason behind this. Our nervous systems are designed to focus on changes in our environment, because changes in our environment are likely due to either threats or opportunities. A threat could be something that could hurt you, like you hear a creaking branch of a tree and it might fall on top of your head. Um, or you hear all of a sudden a weird animal noise and you have no idea what animal that is. Or it could be an opportunity where all of a sudden you smell a food that someone's baking a pie and it's an opportunity to maybe get a piece of pie. Um, but after you've gone through the initial assessment of identifying what that sensory stimulus was, figuring out which tree branch it is, and you look at it to inspect it to see if it's dangerous, or you go into the kitchen and you see if you are smelling an actual pie coming out of the oven. After you've assessed your environment, you can discard that information. It's no longer novel, and you would have protected yourself from a threat and taken advantage of your opportunity, so it's no longer needed. So our nervous system begins to ignore it. For an endocrine response, it's very different. When you secrete a hormone, that signal persists. For example, the release of growth hormone. In puberty especially, preteens start releasing a huge amount of growth hormone that encourages their growth spurt to adulthood. This hormonal response persists for years until they have reached their full adult height. This signal does not extinguish. There is no adaptation to the stimuli, and the body continues to grow as long as the growth hormone signal persists. Another difference between the applications of the nervous and endocrine system is the area of the effect. If a gland is secreting a hormone, it is entering the bloodstream and is distributing everywhere that blood is distributed, which is pretty much throughout the body, with very few exceptions, such as the lens of the eye, for example. Therefore, any cell that can respond to that hormone signal will. So it's a very general broadcast message with widespread effects throughout the body and many organs. The nervous system targets its response. If you want to move a particular muscle, only that muscle is sent an action potential through a motor neuron to activate its neuromuscular junction. And only those muscle fibers will contract. So there's a very restricted and focused area of effect. So there other things that are very similar between the nervous system and the endocrine system. For example, the nervous system and the endocrine system share chemical signals. There are certain compounds that function as both hormones in the endocrine system and as neurotransmitters. Examples of this are norepinephrine, otherwise known as noradrenaline, and dopamine. Another similarity is that some neuroendocrine cells or neurons release hormones into the bloodstream. For example, oxytocin is released by neuroendocrine cells and catecholamines. The nervous system and the endocrine system obviously frequently have overlapping effects on the same target cells. So there are cells that can respond to nervous system signals and hormonal signals. For example, glycogen hydrolysis in the liver responds to norepinephrine, which is a fight or flight signal, and glucagon, which is a hormonal signal. And the two systems regulate each other. Neurons trigger hormone secretions, and hormones stimulate or inhibit neurons. So that's the basic compare and contrast of the nervous system and the endocrine system. But now let's also compare endocrine glands, which produce hormones, to exocrine glands. You should recall the discussion, an introduction of exocrine glands when we were discussing the integumentary system in Bio 11 in the first semester of anatomy and physiology. Exocrine glands have ducts, and they are secreting things to an external surface. Now, be careful with this because the human body is basically a tube, kind of like an earthworm. And if you were to swallow an apple seed accidentally, okay, and then the next day you go to the bathroom and the apple seed enters the toilet bowl, that apple seed never actually entered your body. It just passed through it, kind of like a straw could pass through the hole of a donut, okay? It never actually entered the donut. In order to enter the donut, you would have to break the donut open and reach the crummy part inside. So I mention this because this helps define what an exterior surface is. Anything lining your gastrointestinal tract, anything that apple seed would have bumped into while it passed through your body, is considered an exterior surface. So all of the glands that are producing digestive fluids and mucus in your digestive system are exocrine glands. They are glands that secrete liquids through ducts to the surface of the mucosa of the digestive tract. As similarly to the mucosal layer, the epithelium in your respiratory tract. Endocrine glands don't have ducts. Endocrine glands instead are secreting their chemical products directly into the bloodstream. So instead of a duct network, they have a very strong blood supply, a very good network of capillaries that they introduce the compound they're producing to the blood through. So these are considered internal secretions. And the secretions are carried away from the glands in through the bloodstream and distributed throughout the body until the hormones reach their target areas. So this is a schematic showing the overall setup of the endocrine system. There's the signaling cell, which is in a gland, an endocrine gland, producing a chemical signal that's released into the capillaries that are around the gland, and that carries that chemical signal through the bloodstream to a remote target cell. So the endocrine system consists of the glands and tissues and cells that secrete these hormones. And they are obviously intimately related to the circulatory system that is distributing all of the hormones. And the hormones themselves are the chemicals, those chemical messengers that are transferred through the bloodstream to target tissues. So what happens when a cell is exposed to a hormone? Well, in order for a cell in a target tissue to respond to a hormone, it must have an appropriate hormone receptor. A receptor is a protein or a glycoprotein that is either found on the surface of the cell or inside the cytoplasm of the cell or in the nucleus of the cell. And the hormone is able to reach this receptor and bind to it. And one of the things that you are hopefully told in Bio 11 and taught is that when something binds to a protein, the protein changes shape. So when a hormone binds to a hormone receptor, the receptor will change shape. And when that receptor changes shape, all of a sudden that protein is able to do things that it couldn't do before. And those things are what changes the behavior of the cell and creates the effect on the target cell. But the number one thing to remember is, in order for a cell to respond to a hormone, it has to have a receptor that will bind to that hormone. So in this case, in this green cell, it has a bunch of these blue receptors that only bind to hormone A. And this blue cell over here also has those receptors. So it is also sensitive and would respond if hormone A was released in the blood. But it also has a second receptor that responds to a different hormone, hormone B. Then finally, this red cell only has receptors for hormone B. If a person is only producing hormone A, this green cell will respond, the blue cell will respond, but the red one will not. And the reason why the red one will not is because it doesn't have a receptor for hormone A. On the flip side, if instead the person is not producing hormone A, they are only producing hormone B, the red cell will respond, the blue cell will respond, but the green cell will not because it lacks a receptor for hormone B. The only cell that is capable of responding to both signals is this middle blue cell because it possesses the hormones for both. Usually, a target cell has a few thousand receptors for a given hormone. So, receptor hormone interactions exhibit specificity and saturation. The specificity I explained on the previous slide. A cell needs to have the proper receptor that reacts and binds to the hormone being released. It also has saturability because there's usually a few thousand of these receptors on the surface of a cell. And there's a maximum number of hormone molecules that can bind to the cell. There's a certain point where every receptor is bound to a hormone. When that case happens, we have the maximum response. The cell cannot respond any stronger to this hormone because every receptor it has is bound and activated. But frequently, only a fraction of these receptors will actually be bound to the hormone because not much of the hormone exists. So there's an ability to have a graded response. With increasing amount of hormone, you will have a larger response. Now let's start talking about the types of hormones. There are two main types of hormones. They can be categorized as hydrophilic, which means water-loving. These are hormones that are able to dissolve in blood and water and extracellular fluids. And then there are hydrophobic hormones. Hydrophobic hormones are unable to directly dissolve in plasma without the help of, proteins or something else to keep them in solution. These two types of hormones behave very differently in the body because they move around the body and bind to things very differently. Hydrophilic hormones freely diffuse in the plasma and extracellular fluid and any place where there's a watery matrix. Hydrophobic hormones prefer to be situated inside a membrane, or they are bound to a protein that is designed to carry fatty things. So they move about the body very differently, and they have different features of a cell that are viewed as a barrier versus an area where it can freely move about. So let's first talk about hydrophilic hormones. The primary category of hydrophilic hormones are amine hormones. And of course, I have to start off with an exception to the rule. Amine hormones include a lot of hormones that are synthesized from the amino acid tyrosine. And the hormones that are produced by tyrosine include thyroid hormones and the catecholamines dopamine, epinephrine, and norepinephrine. Right off the bat, the first thing I want to point out is that thyroid hormones are not hydrophilic. They're actually hydrophobic. But I put them here because they are made from tyrosine, okay? The purpose of thyroid hormones is they regulate the metabolism. We'll talk about all the features of hydrophobic hormones like tyrosine in a few slides. But many other tyrosine-derived hormones include catecholamines, which are legitimately hydrophilic. And these hormones include dopamine, which inhibits the release of some anterior pituitary hormones, and epinephrine and norepinephrine, otherwise known as adrenaline and noradrenaline, which are responsible for your fight or flight response. Down here, for the people who love biochemistry, I provided the synthetic pathway of these tyrosine-derived hormones. You do not have to memorize this pathway down here for an exam, but I put it here just for the sake of completeness and just so you understand what I mean by a tyrosine-derived hormone. Here is the amino acid tyrosine. Through a series of enzymes, here's the first, the second, the third, and the fourth, these hormones sequentially convert tyrosine into different products that eventually produce different hormones. So tyrosine hydroxylase will modify the tyrosine to produce dihydroxyphenylalanine, which is otherwise known as DOPA. And then we have an aromatic amino acid decarboxylase, which will convert the DOPA into dopamine. And then dopamine beta monooxygenase will convert it into norepinephrine. And then phenylethanolamine N-methyltransferase will then convert the norepinephrine to epinephrine. Once again, you don't have to memorize this, but it just gives you an idea of the synthetic pathway and the origins, the tyrosine origins of these several different hormones. Another group of hydrophilic hormones that are amine hormones synthesized from tryptophan, which is another amino acid. And a great example of that is melatonin. You may have heard of melatonin because a lot of pharmacies sell it. And in general, what are people these days taking melatonin for? To help them go to sleep. Okay. But what is melatonin really? It's a hormone that is produced by a teeny gland called the pineal gland, which regulates your circadian rhythm. When the sun sets and light levels get low, your pineal gland will start producing melatonin, and the melatonin levels will rise higher and higher, and this is the structure of melatonin, until you start to get quite sleepy and you fall asleep. And the melatonin levels will only drop after you've been asleep a while. People frequently take synthetic melatonin in order to encourage themselves to fall asleep when they have sleep regulation issues. Melatonin levels obviously are also very much involved in people who have jet lag because they had to travel from one time zone to another. In addition to the amine hormones, we have peptide hormones. And instead of them being derived from a single amino acid, they're being derived from short chains of amino acids. Examples of peptide hormones include antidiuretic hormone, which is a hormone that's important in fluid balance. You're going to become very familiar with this hormone as the semester progresses. And atrial natriuretic peptide, which is another hormone that helps regulate blood pressure. Protein hormones are basically derived from things that are longer than short, they're longer polypeptides instead of short chains. Examples of this include growth hormone and follicle stimulating hormone. Growth hormone is quite obvious. It stimulates the growth of new cells. Follicle stimulating hormone is produced during puberty and adulthood and promotes the formation of eggs and sperm. In this first portion of the unit, I've been concentrating on the types of hormones, hydrophilic versus hydrophobic, because they behave very differently in the body. Hydrophilic hormones, hydrophilic means water loving, can freely dissolve in anything watery. So the plasma is a perfect place for it to dissolve and transport through the blood. Extracellular fluid is watery, so it can freely move about over here. But once it reaches the membrane surface, this is an impenetrable surface because it is lipophilic. It is made up of lots of phospholipids. Therefore, it cannot freely diffuse across the plasma membrane and move intracellularly. So this is the barrier, the limit of the movement of hydrophilic hormones. Therefore, hydrophilic hormones will activate cells by binding to receptors on the surface of the cell. And this allows a conformational change in this protein that will change the shape of the protein on the inside of the cell, on the inner leaflet of the membrane. And that will allow it to activate G proteins that are associated with the receptor. The G protein will activate an enzyme called adenylyl cyclase. And what adenylyl cyclase does is take ATP, adenosine triphosphate, and convert it into cyclic AMP. And cyclic AMP will then do a host of things inside the cell. It will start activating a bunch of protein kinases. And these kinases are going to add phosphates to a bunch of proteins. And these proteins are going to become activated and they are going to start doing activities in the cell that they couldn't do before the adenylyl cyclase was activated. So this will change the overall behavior of the cell. We are turning on and off the activity of various proteins that are normally found in the cytoplasm. Now, why bother with all of this? Look at this. We've got an activation of a receptor, then an activation of an enzyme, and then we have cyclic AMP activating a bunch of kinases, which then activates a bunch of proteins. Why not instead erase all of this middle junk and just activate a receptor, and that receptor directly activates the protein? What could be the benefit of all these intermediate steps? signal amplification. That's what the benefit is. Let's say one molecule of a hormone will activate one receptor. That one receptor has the ability to activate many cyclic AMP and protein kinases because it's activating adenylyl cyclase that will really turn on cyclic AMP production. Okay? And when we have many protein kinases activated, you can have a very large quantity of intracellular enzymes activated and intracellular molecules. And they are going to be churning continually, producing even larger quantities of product that will produce a huge effect. If we eliminate all these middle steps, we would have a very small, trivial effect from the activation of one hormone receptor. But because we have all these intermediate steps, we can have a very profound, enduring effect. So that is the purpose behind all these middle layers. Now, I should mention that you are not required to, memorize the specifics of any specific cyclic AMP pathway. I do expect you to remember the basic steps that I presented on the previous page. Now let's discuss hydrophobic hormones. And if you recall, when I introduced hydrophilic hormones, I started off with the exception, the one tyrosine-derived hormone that is hydrophobic, and that is thyroid hormone. But in addition to thyroid hormone being hydrophobic, we have multiple steroid hormones that are derived from cholesterol. And if you recall from Bio 11 and the biochemistry unit of Bio 11, cholesterol is predominantly made out of carbon and hydrogen. It has a little bit of oxygen, not much, and it has a lot of nonpolar covalent bonds, so it's very hydrophobic. So steroid hormones, because they're derived from cholesterol, are not soluble in water. So that means the cytoplasm of the cell is a barrier to it. The extracellular matrix is a barrier. The plasma, the blood, is a barrier. However, the cell membrane is not, because it's made out of a lot of phospholipids. So steroid hormones are able to easily pass through membranes, but they need help moving around in aqueous compartments like the plasma, the extracellular fluid, and the cytoplasm. Usually they're bound to binding proteins that have little hydrophobic pockets to help carry them around in these fluids. So here are some examples of hydrophobic hormones, and this is the parent molecule they're derived from. This is cholesterol. Cholesterol, without even staring at the details, the minutia of the molecule, it's very obvious what cholesterol is because it has these four rings, these three hexagonal rings and this one five-sided figure off on the end. So all of these look fairly similar with these four ringed structures. Sometimes they're modified a little bit. Double bonds appear in random places, but overall they are all distinctly familiar cholesterol-like structures. Steroid hormones include the sex steroids such as testosterone and estrogens. Also cortisol, which is produced by the adrenal glands and is a stress hormone, and aldosterone. But obviously the pathway of before with a G protein coupled receptor and adenylyl cyclase is just not a reasonable signaling pathway for a hydrophobic hormone because the membrane is not a barrier to it. All these hydrophilic fluid compartments are. So a lipid soluble hormone will likely be trafficked with a binding protein or something through the plasma. It will also be trafficked through the extracellular fluid, and then it will be deposited on the surface of the membrane. It will very easily cross the membrane without any assistance. It doesn't need a channel or anything to take it up, because it will just, fat dissolves in fat. It will just freely enter the membrane without any difficulty. However, it will be difficult for it to leave the membrane and enter the aqueous cytoplasm. So it will have to bind a protein, which we will call a receptor, in the cytoplasm and form a receptor hormone complex. This receptor hormone complex will float across the cytoplasm, enter the nucleus, and directly interact with the cell's DNA. And there it will trigger gene transcription, and that will influence what proteins are made in the cell. And by influencing what proteins are being made in the cell, we are going to directly influence the cell's activity. Oh, if you ever decide to take a biochemistry class, be forewarned. This is the kind of thing that you would be expected to memorize in a lecture. You don't have to memorize this picture, okay? It's merely there to provide you with an idea of what happens when a certain hormone binds a certain receptor. Um, Gene regulation will cause many different things to happen. Um, and why? Well, I think right now it's a good time to pause on our discussion of hormones, and instead just take a step back and discuss um, how cell functions are regulated overall, and what role does protein synthesis play in the regulation of cell functions. Um, I know for a fact that there is no prerequisite course for Bio 12 that requires you to study protein synthesis. So that means some of you probably already learned about protein synthesis. Perhaps you took a general biology class and they presented it, but others of you may have not. So I would like to pause right here and provide you with the information on protein synthesis so all of you are on the same page and understand how protein synthesis affects the function of a cell. So we're going to review what DNA is, what genes are, what protein synthesis is, and how that affects cell behavior. The first thing to know is that cells are constantly replacing proteins. It's not like a protein is made once and it stays in that cell forever. Cells, proteins are very much like cell phones or a pair of jeans or whatever, a t-shirt. They're great at first, but as time goes by, they get a little worn out. They start to get a little quirky. They stop functioning perfectly well. You know, your t-shirt might get a hole in it. It might, start to get really floppy and misshapen. Your cell phone may start to get a little weird and quirky, and it no longer functions the way it does. It drops calls very often. You miss text messages and whatever, screen cracks, whatever, and eventually you have to replace all these things. It's a fact of life. Nothing lasts forever. It's the same for a cell. Proteins need to be replaced, and they need to be replaced with reasonable regularity. They just start getting a little beat up and not functioning properly. So what the cell does is it sends them to a lysosome to dissolve the old proteins, it replaces it by making new ones, replacement proteins. So all the enzymes and structural proteins that you have in your body and all of your receptors, they're being continually recycled and replaced. The old ones that are worn out are gotten rid of and broken down, and new ones are made and put into action. So by changing what proteins are expressed in a cell, the activity of the cell can be changed. What I mean by that is, let's take, for example, hormone receptors. Hormone receptors are proteins. If the cell makes a lot of a hormone receptor and puts thousands of them on the surface, when that hormone is released, the cell is going to be very sensitive to that signal, and it's going to respond greatly to that hormone signal and change its behavior. If instead it stops making those receptors and the old ones are taken away because they're worn out, but they aren't replaced with new ones, next time that hormone signal comes by, the cell is not going to respond very well to it because it doesn't have many hormone receptors at all. If it has no hormone receptors, it won't respond at all. So by changing what proteins are expressed in a cell, the activity of the cell is changed. And herein lies the potency of hydrophobic hormones. Hydrophobic hormones directly influence what proteins are being made by a cell. It will tell a cell to make more of a receptor or less of a receptor, more of an enzyme or less of an enzyme. And that is greatly going to affect how a cell behaves. So let's start off with what a protein is. And since we're discussing hormones, I might as well give an example of a protein that's a hormone. So I chose the very famous hormone, insulin. Insulin is a relatively simple protein. It is two chains. There's an alpha chain and a beta chain, and they're connected by a couple disulfide bonds. Um, And what you can see here in this schematic, this cartoon, they drew it as if each amino acid is like a bead. So it's almost like two beaded chains connected together. Well, all proteins are basically similar in their basic structure. They are made up of amino acids, which you could draw as beads, linked together into chains. There are 20 possible amino acids to be used to make a protein. So you could almost imagine it as 20 different colored beads. And they are linked together in a specific sequence that is unique for each protein. So insulin has this specific sequence: glycine, isoleucine, valine. You get the idea. And you read through each individual one. It's kind of like having a necklace which starts with a green bead, a blue bead, a yellow bead, an orange bead. You get the idea. And if you are assembling a new protein and building a new protein, the instructions you need for assembly is, first you start off with the blue bead, then you move to the green bead, then you go to the yellow bead, and then you go to the orange bead, and you just follow the colors and the sequence and make the chain the proper length with the proper sequence of colors. When a ribosome is making a new protein, it does the exact same thing. It follows the instructions on which amino acid to put in which order, and they make a chain of amino acids of a specific length. That is the proper sequence. So the analogy, that's why I have a picture of many beaded bracelets here. So building proteins, the instructions to build a protein is very similar to the instructions to build a necklace. Specify what type of bead and specify the pattern, the sequence, and the length, how many beads overall. Is it blue, yellow, blue, yellow, blue, yellow? Okay, how many beads at all? 50? Cool. Now you know how to make that particular necklace. And that's exactly the same kind of instructions you would give a ribosome to make a particular peptide chain that gets folded ultimately into a protein. So a protein can be short with less than 50 amino acids, or it can be very long with more than 500 amino acids. And the uniqueness of each type of protein is in the length of the chain and the sequence of the amino acids. So once again, in order to make a specific protein, a ribosome needs to know the length of the peptide chain and the sequence of the amino acids that make up the chain. And then while it builds the new peptide, it just has to follow the instructions and put them in the correct sequence and make the correct length. Where do they get those instructions from? Well, you've inherited it from your parents. It's in your DNA. A gene is a coded segment of DNA that provides instructions on how a ribosome should make a specific protein. In other words, all the inherited information you have from your parents on how to be a human being is merely a series of recipes to make all necessary human proteins. And each individual protein recipe or gene just gives a ribosome information on the sequence of the amino acids and the length of the chain to make each individual protein. Now, how does DNA do this? Well, it's a very special macromolecule that has a pattern of nucleotides. There are 4 nucleotides. I will simplify them by just calling them A, C, T, and G. And they are in a specific sequence to create a recipe for the gene. You may recall from the cell unit from Bio 11 that DNA stays in the nucleus. It doesn't leave. Where are ribosomes again? No, yeah, they're in the cytoplasm, free floating, or maybe they're attached to the rough endoplasmic reticulum. Well, how does that work? If the information on how to make a protein is stuck in the nucleus, but the machinery that makes the proteins is in the cytoplasm by the rough ER, well, we have to make a copy of the recipe that is able to leave the nucleus and go to the cytoplasm. This copy is called RNA. It's messenger RNA or mRNA. The process of protein synthesis is the following. In the nucleus, we have DNA, and it has the information on how to make every protein a human cell could possibly need. Every protein in the human body. In order to make more, for example, dopamine receptors, we have to look up and find the part of a chromosome that has the dopamine receptor gene. And just that tiny segment of that chromosome, we transcribe it. We make a copy of it. And that is an mRNA copy of only, for example, the dopamine receptor gene. That mRNA copy is able to leave the nucleus and enter the cytoplasm where the ribosomes are. The ribosomes will latch on to the mRNA, read the instructions on how to make new dopamine receptors, and translate those instructions and make a peptide, a chain of amino acids that can be folded up to make a new protein, a new dopamine receptor. So it's a 2-step process. The first step for protein synthesis is transcription, where we find the gene and make a copy of only the information on how to make the protein we need, and that mRNA copy is then translated, and the ribosomes read it and make the protein. Where does hormone regulation come into play with this? Well, hydrophobic hormones directly influence this step right here. It directly influences what mRNAs are made and how many copies of each mRNA are made. If you make a lot of mRNA copies of the dopamine receptor, your ribosomes are going to make tons of dopamine receptors, and the cell is going to become increasingly sensitive to a dopamine signal. If instead you decide to make much less of an enzyme, for example, you decide to make much less adenylyl cyclase, well then your hydrophilic hormones are not going to exhibit that much of a response anymore to the cell. They're not going to affect the cell very much anymore because all G protein coupled receptors are not going to be able to make much cyclic AMP because you are made less of the enzyme that makes cyclic AMP. So hormones can greatly regulate the behavior of cells by influencing the amount of proteins that are found in the cells. So after the transcription is made, the transcription is performed and the mRNA is made, we translate the mRNA and produce peptides that are folded to make proteins. And this is a case of amplification of signal. If the hormone influences you to make an extra 30% of an mRNA, you have 30% more dopamine receptor mRNA, you are going to get much more than 30% more dopamine receptors because each mRNA is reread by ribosomes multiple times. So there is an amplification of the signal. So this is a slightly more in-depth slide that explains the first phase of protein synthesis, which is transcription. And this is for eukaryotes because we are eukaryotes. In the case of transcription, it begins with initiation, where if you stimulate a gene to be transcribed, the DNA where the gene is located, the two strands will separate. An RNA polymerase will latch on and start copying, making a complementary strand of pre-mRNA that matches the DNA sequence. So it's making an mRNA copy of the gene. And this happens, the initiation is where the RNA polymerase latches on and the two strands of DNA separate. Elongation is when this copy is being made. Once the copy has finished, the RNA polymerase reached the end of the gene, it will terminate, it will add a poly-A tail, and this RNA can now float out of the nucleus and enter the cytoplasm for translation. For the process of translation, the mRNA leaves the nucleus and will bind a ribosome. The ribosomes will read a codon at a time and translate the information provided on the gene to make an amino acid sequence, which is sort of like translating from one language to another. To understand the mRNA information, you have to understand the language of genes. So I already mentioned that DNA has 4 different bases, A, T, C, G. RNA the RNA copy that is made during transcription is similar. It is A, C, G, and U instead of T. But it's kind of like having an alphabet with 4 letters to simplify this. When ribosomes are reading an mRNA, they read 3 bases at a time. That's kind of like reading a 3-letter word. So you can think of the language of genes as being a language that has 4 letters in the alphabet and 3-letter words. So there are 64 possible codons because there's 64 possible words if you were to reorganize those 4 letters around and made 3-letter words from it. So these 64 possible codons represent the 20 amino acids. So now all you have to do is write AUG. Well, what does AUG equal? Well, AUG is the start codon, and it equals methionine, which is the first amino acid. So when the ribosome reads AUG, it knows, oh, I have to add a methionine. So this is the way it reads. It reads GCU, and that's the first word. And the ribosome knows that that means alanine. And ACG is the second word, the second codon, and that equals threonine. And then it moves down and reads GAG, which is the third codon, which equals glutamate. And it's building a chain of amino acids. So for every 3 bases, this is codon 1, codon 2, codon 3, each one equals a single amino acid. And the peptide slowly builds until it reaches a codon like this, UAG, which is a stop codon. It tells the ribosome to basically unlatch from the mRNA and stop translating. So now this chain floats away and becomes folded and becomes a protein. So each mRNA can be read multiple times by many ribosomes. We could have one ribosome over here and another ribosome down here. So you can make multiple copies of the protein from one mRNA. Now, mRNAs don't last forever. By design, they're very fragile and will only last a certain period of time. So in order for a cell to keep making tons of this protein, it has to make new copies of mRNA. This is very important because if you have a start signal where you produce an mRNA, you have to be able to turn off that signal and stop making the protein. So the mRNA gradually degrades and the protein will stop being made at that point unless the cell restarts the process with new mRNA. So what does this look like with the interaction between the ribosome, tRNAs, and the mRNA that was produced in the nucleus? Let's look at this a little bit closer. An mRNA leaves the nucleus. It floats into the cytoplasm, and it meets a ribosome. A ribosome has 2 subunits. Here it is pictured in light green and a darker green. It clamps on to the mRNA, kind of like a clamshell closing. And it has 3 sites. It has an A site, a P site, and an E site. In the A site, a tRNA, which is a special RNA that has an amino acid attached to it, will enter and see if it can bind to the mRNA. It has an anticodon site over here. It's almost like a lock and a key. If this can bind here, that means it has the proper amino acid and it can transfer to build the peptide chain. If instead it doesn't fit against this codon, these 3 bases, that means it has the wrong amino acid and it will float out of the A site and allow a new tRNA to come in. So if this tRNA can interact and bind to this codon down here, what will happen is it will transfer its amino acid to the growing peptide and then shift to the P site. In the P site, the tRNA is empty. It no longer has its amino acid, and it will then exit through the E site as an empty tRNA that needs to be reloaded with an amino acid. After this happens, the ribosome will shift downwards, and there'll be a new codon in the A site, and a new amino acid attached to a tRNA will enter the A site and transfer to the growing peptide. And it will continue to move downwards, down the mRNA, and continue to translate one codon at a time, every 3 bases. So let's look at this in closer detail. In the ribosome, the tRNA has a specific amino acid associated with it. This amino acid, it matches the proper anticodon over here. These anticodons bind to codons. A always binds to U. C always binds to G. So whatever the anticodon is here the codon on the mRNA is UUU. So very simply, we need AAA here. This tRNA will be able to interact with this codon, which means that this amino acid, it will be able to transfer to the peptide. And then after this happens, the tRNA moves to the E site and then exits the ribosome. So this is how the codon-anticodon pairing occurs and how we select the proper amino acid to add to the chain. And once again, once the amino acid is transferred in the P site, it leaves, the empty tRNA leaves through the E site. And the ribosome keeps shifting downwards, translating each codon in sequence, and the amino acid chain grows. And this is something for you to read on your own. It just basically reiterates exactly what I explained. I just presented in 2 forms in order for some people who didn't like the previous graphics, you can read it on this graphic. So how does this protein synthesis allow for the regulation of cellular activities? Well, genes are turned on and off from day to day. And by regulating how much RNA a specific gene is made every day, the cell regulates how much protein of that particular type is made by the ribosomes every day. And ultimately, the abundance of that particular protein in the cell. That dictates whether the cell can perform a specific function or not. So the regulation of gene expression is definitely beyond the scope of this class, but I just wanted to explain to you the mechanism, the basics of the mechanism of how hormones actually regulate the activity of a cell. It's not magic, okay? Without this explanation, it would seem kind of magical and, like beyond the scope of understanding. No, it's actually, it's well understood. It's just complex and, in the realm of geneticists and molecular biologists. So ultimately, steroid hormones influence which genes are being turned on or transcribed at any given moment. And that regulates what proteins are present in the cell and what the cell is capable of doing. So, to review the first portion of this lecture, hormones are released by glands and travel through the blood throughout the body. A hormone will activate any cell that has an appropriate hormone receptor for it. The hydrophilic hormones have receptors on the cell's surface because the cell's membrane acts as a barrier. Hydrophobic hormones will generally have intracellular receptors that shuttle the hormone through the cytoplasm into the nucleus where they affect gene transcription. And due to the signal amplification in both types of hormone signaling, it only takes a tiny amount of hormone to cause a large cellular response. Once again, hydrophobic hormones freely diffuse across a membrane but require a hormone receptor complex for movement across the watery cytoplasm, which will take it into the nucleus where it will directly interact with DNA. And hydrophilic hormones, the barrier is the membrane. So the receptor will be sitting on the surface of the membrane, and that will result when it binds in a shape change that causes different proteins in the cytoplasm to be able to interact with it. For example, adenylyl cyclase that will cause all these second messenger effects that will cause a huge change in cell activity in the cytoplasm. So now we are moving on to the specifics of endocrinology. We're doing an overview of the major glands and the hormones that they produce. Here is a diagram showing the location of the major glands we're going to discuss. There's the pineal gland, the hypothalamus, and the pituitary, the thyroid and the thymus, the adrenal glands, which sit on top of your kidneys, the pancreas, the ovaries, and the testes. The first I'd like to go through is the pineal gland, which is attached to the roof of the third ventricle of the brain. It is quite small in adults, but has a very profound function. Its purpose is to synchronize your daily physiological activities with a 24-hour rhythm. In other words, synchronize us to sunlight. When the sun sets, now keep in mind, we evolved without electricity, okay? So when the sun sets, everything goes dark and it's nighttime. We're supposed to be falling asleep when the sun sets and waking up when the sun rises. So with that concept in mind, when the sun sets, the pineal gland starts secreting a lot of melatonin in response to the darkness that's surrounding us. The increased levels in melatonin cause us to feel sleepy. We eventually fall asleep. Melatonin levels don't drop until we've been asleep for a certain amount of time, and by the time we have sufficient sleep and our melatonin levels have dropped enough, the sun is coming up and we wake up. So the pineal gland's function and melatonin's function is to synchronize us to the daylight patterns in our environment. You may be familiar with melatonin because many people buy it at the pharmacy, and pop a little melatonin in the evening to help them feel sleepy so they fall asleep more promptly. One of the reasons why in modern society we have problems falling asleep promptly is because we are looking at screens that emit a lot of blue wavelength light that actually causes the pineal gland to not secrete melatonin. So I propose to you, if you want to fall asleep and have a decent quality of sleep, you shouldn't be looking at screens for a few hours before you go to bed. Or if you have to look at screens in the evening, I suggest you either, some devices have a blue light function where it eliminates the blue light so it doesn't keep you awake. Otherwise, you can invest in a very cheap pair of blue light blocking glasses. You can get them online for probably $10, and they will help you fall asleep despite the fact that you have to look at a computer screen before bedtime. The next gland that we're going to discuss, this is a biggie. It's the hypothalamus. It is located just beneath the thalamus of the brain, hence the name, and is responsible for regulating water balance, thermoregulation, sex drive, childbirth. But many of the functions that are carried out by the hypothalamus are truly executed by the pituitary gland. There's a very intimate relationship between the hypothalamus and the pituitary. And that intimate relationship is through the hypophyseal portal system. Basically, what happens is there are a lot of hormones that are released by the hypothalamus that travel through the hypophyseal portal system and go directly to the anterior pituitary. And then the anterior pituitary changes its behavior and what hormones it releases in response to what the hypothalamus dictates. So there are 8 hormones that are produced by the hypothalamus. 2 are released in capillaries in the posterior pituitary when hypothalamic neurons are stimulated, and 6 are either directly stimulating or inhibiting the anterior pituitary. And here is a list of hormones that the hypothalamus and the pituitary are producing. Unfortunately, yes, you do have to know all of these. What I strongly suggest everyone does is take a piece of paper and you write down the name of the gland, list all of the hormones, and list what they do. Okay? And then also mention whether they're hydrophobic or hydrophilic, if that information was provided. So the hypothalamus has a tendency of producing hormones that influence the effect of the pituitary gland. For example, you might see here that the hypothalamus produces growth hormone releasing hormone. When this growth hormone releasing hormone is produced, it stimulates the pituitary to make growth hormone. So you see how that works? So the pituitary gland is suspended beneath the hypothalamus and is composed of 2 structures. There's an anterior pituitary, which is otherwise known as the adenohypophysis, and the posterior pituitary, which is really a downgrowth of the brain and is otherwise known as the neurohypophysis. The adenohypophysis is the anterior 3/4 of the pituitary, and it's linked to the hypothalamus through that hypophyseal portal system that I discussed earlier. So there are capillaries in the hypothalamus that are directly connected to capillaries in the adenohypophysis by portal venules. So the hormones produced by the hypothalamus are directly sent to this structure. The neurohypophysis, otherwise known as the posterior pituitary, is nerve tissue. It's not really a true gland. There are nerve cell bodies in the hypothalamus that pass down the stalk and end in the posterior lobe. The hypothalamic neurons secrete hormones that are stored in the posterior pituitary and released into the blood. So the anterior pituitary synthesizes and secretes 6 major hormones. I provided the acronyms. I detest acronyms, but I provided them just because some places on the internet, some sources in the literature really love using acronyms, so I feel like you should be familiar with them, but I heartily encourage people to write things out because when you have these little abbreviations, frequently the same abbreviation means different things to different people. Case in point, I used to work in Brookhaven National Labs, so to me the DOE is the Department of Energy. But for some of my colleagues who used to work in the New York City public schools, the DOE for them is the Department of Education. So this is why I don't like abbreviations. I don't like acronyms very much. Nonetheless, I provide them just in case you bump into them. So the first of the anterior hormones I would like you to know is thyroid stimulating hormone, which obviously stimulates the thyroid gland to produce thyroid hormone. They're also follicle stimulating hormone and luteinizing hormone, which are gonadotropins. They are sex hormones that stimulate the secretion of other sex hormones in addition to the production of gametes. And then there are a bunch of other hormones. Some you will find pretty familiar, like growth hormone. That's kind of self-explanatory. It stimulates mitosis and cell differentiation. And then some other weird hormones that right now you may be unfamiliar with, but by the end of the course, hopefully you will be very familiar with them, including prolactin. Prolactin is what stimulates lactation after childbirth, the production of milk, and adrenocorticotropic hormone, which stimulates the adrenal cortex to secrete its glucocorticoids, which will be coming up in a future slide. Um, some posterior pituitary hormones include antidiuretic hormone. This is a hormone that encourages water retention when you're somewhat dehydrated and the maintenance of blood volume. And oxytocin, which is a feel-good hormone which surges during sexual arousal and also during childbirth because it stimulates labor contractions during childbirth. It also helps with the emotional bonding between baby and mom during childbirth and also during breastfeeding, and stimulates lactation. Um, So it's a multi-pronged hormone. Thyroid gland. The thyroid gland most famously regulates your overall metabolic rate. It does so by secreting T4 and T3. Ultimately, T4 is converted to T3, and T3's function is to increase your metabolic rate, increase your oxygen consumption, increase heat production, appetite, increase growth hormone secretion, and alertness. In addition to that, the thyroid gland has another function. Parafollicular cells secrete calcitonin. Oh yeah, calcitonin, I remember that. It's from Bio 11 in the skeletal unit, right? So if you were to eat a meal that was really rich in calcium, say you had a meal that had tons of cheese in it, and with a pile of broccoli on the side, you just ate a boatload of calcium and your blood calcium levels are very high. Your parafollicular cells will secrete calcitonin, and that's going to stimulate osteoblasts to deposit more bone, more hydroxyapatite, if you recall from that unit. So you can store that calcium for later use. Parathyroid glands. Parathyroid glands are these four little dots right here sitting on top of the thyroid. Um, Once again, if you harken back to your Bio 11 skeletal unit, The parathyroid hormone increases blood calcium levels. So when your blood calcium levels drop because you aren't eating any cheese or broccoli at the moment, it will synthesize calcitriol, which increases absorption of calcium and decreases secretion in the urine of calcium, and increases the breakdown of bone to maintain your blood calcium levels. Thymus gland. The thymus gland, it plays a role in multiple organ systems: the endocrine system, the lymphatic system, and the immune system. It's the site of T cell maturation. This is incredibly important for your acquired immunity, and we're going to be discussing this extensively in the middle of the semester. The thymus also secretes hormones that stimulate the development of other lymphatic organs, and also stimulates the activity of T lymphocytes. The adrenal glands. The adrenal glands look like little hats sitting on top of your kidneys. They are retroperitoneal, which means they are right against the back wall of the abdominal pelvic cavity, right where the kidneys are. And they are made up of two tissues, an adrenal cortex and a medulla. Here's the histology of the adrenal gland. The inner section is, of course, the medulla, and the more superficial section is the cortex. The medulla has a dual nature. It acts as an endocrine gland and a sympathetic ganglion, which responds to the sympathetic nervous system. So when it's stimulated, it releases neurotransmitters into the bloodstream, such as the catecholamines epinephrine and norepinephrine, and also dopamine. The cortex is the superficial tissue layer surrounding the medulla, and it produces a whole lot of steroid hormones called corticosteroids. Um, so the inner cortex increases our alertness. It has an effect that's longer lasting than neurotransmitters. We're preparing for a fight or flight reaction. So we're mobilizing our energy, our fuels, fatty acids, glucose, boosting our glucose levels, inhibiting insulin secretion, increasing our blood pressure and heart rate. We're getting ready to flee from a potentially threatening situation. And simultaneously, um, we're suppressing the rest or digest scenario, where digestion and neuroproduction are reduced in order to promote and save our energy for the fight or flight. The corticosteroid categories for the cortex include mineral corticoids, glucocorticoids, and sex steroids. The sex steroids are obviously the androgens and estradiol. The androgens are the male hormones, but they are found, both androgens and estrogens are found in low levels in both genders. It's just the male hormones, the androgens are higher in men and the estrogens are higher in females. Um, Androgens set the libido throughout life, and it plays a large role in male development. Estradiol is in small quantities in men, but uh, uh, plays a major role in women, especially after menopause, for sustaining adult bone mass. Um, Glucocorticoids regulate the metabolism and are fuel sources. Cortisol is a stress hormone that stimulates fat and protein catabolism, gluconeogenesis, and the release of fatty acids into the blood. And it helps us adapt to stress and repair. One thing I should point out is you have to think of what our bodies are adapted for. The vast majority of human history we were in hunter-gatherer societies, and when we're talking about stress responses, the stress was probably famine. That was the number one reason for a stressful situation. So the stress responses tend to conserve energy in order to ride out a period of time where food was scarce. Um, so that's why cortisol does these things. It's very different from modern stresses with jobs and familial issues, etc. So mineral corticoids regulate electrolyte balance. Aldosterone, for example, will stimulate sodium retention and promote the excretion of potassium. And you might say, why would we want that? We have too much salt in our diets. Indeed, we do. But throughout human history, once again, if you lived inland, if you lived away from the ocean, you did not have a lot of sodium in your diet. So it was critical that you had some sort of mechanism to help retain precious sodium in the absence of sufficient dietary sources. Um, Now let's talk about the pancreas. The pancreas is a very important organ that's not only involved in endocrine function, but also exocrine. It's also an exocrine gland because it's producing digestive enzymes that's secreted into the alimentary canal. So it has 1 to 2 million pancreatic islet cells that produce hormones, that's endocrine function. And then 98% of the cells of the pancreas are producing digestive enzymes as an exocrine function. As far as the endocrine function is concerned, the most famous hormone it produces, the pancreas produces insulin. So beta islet cells produce insulin in response to increased sugar levels in the blood. When you eat a meal and say you consume some bread or starch, pasta, rice, that glucose will enter the bloodstream and your blood sugar levels will increase. The pancreas will detect this and will secrete insulin, which signals to all the cells of the body that there's extra glucose around. Why don't you take that up so it can be used later on? On the flip side, there's also glucagon. The pancreas produces glucagon when you are in between meals. Let's say it's the middle of the night, you haven't eaten in 6 hours. You still need a certain minimal level of glucose in your blood to keep your cells alive. So glucagon will stimulate gluconeogenesis, which is the production of new glucose from reserved nutrients when you have not eaten recently. So this helps maintain a certain level of glucose in the blood at all times. So adipose tissue and will be broken down. There'll be fat catabolism and the release of free fatty acids um, and the production of new glucose. Additional pancreatic hormones include gastrin. Gastrin is an endocrine hormone that stimulates stomach acid secretion and the stomach churning and emptying when new food has just been swallowed. Pancreatic polypeptide will inhibit the gallbladder contraction and the secretion of pancreatic digestive enzymes when you're in between meals. And somatostatin, it partially suppresses the secretion of glucagon and insulin and inhibits nutrient digestion and absorption to help slow down the process of digestion so it's more efficient. You could categorize the pancreatic hormones according to their effect. Hypoglycemic hormones are hormones that serve to lower blood glucose levels, hence hypo. And that an example of that is insulin. Hyperglycemic hormones are hormones that are designed to raise blood glucose concentration when it gets too low. And that includes glucagon, growth hormone, epinephrine, norepinephrine, cortisol, and corticosterone. And now finally, the last category of hormones are the hormones involved in produced by the gonads, which are the ovaries and the testes. The ovaries and the testes serve as both endocrine glands and exocrine glands because they are producing hormones that are secreted into the blood that have global effects throughout the body, but they're also producing something through whole cells that are emptied directly into ducts rather than into the bloodstream. So the ovarian hormones include estradiol, progesterone, and inhibin. The testicular hormones include testosterone, a bunch of weaker androgens, some estrogen, and some inhibin. For the female hormones, the ovaries um, produce estradiol, which produce a huge feminizing effect to the body. And this estradiol levels are especially high during the first 2 weeks of the menstrual cycle when follicles are developing. After ovulation, estradiols drop and progesterone levels begin to increase. So the second half of the monthly cycle will be dominated by progesterone. And the purpose of the progesterone is to prepare the uterus for the possible conception of a developing embryo. The ovarian follicle and corpus luteum also secrete inhibin, and the purpose of the inhibin is to prevent multiple ovulations. So inhibin suppresses follicle stimulating hormone secretion from the anterior pituitary. Follicle stimulating hormones function is to encourage the development of new eggs. So inhibin prevents new eggs from being developed. This is very important because if a woman is pregnant, for example, she does not want to ovulate again and potentially become pregnant with babies of, developing embryos or fetuses of different ages, because you can only give birth once. Progesterone, along with estradiol, will thicken your endometrium and prepare the uterus for the implantation of a fertilized egg. The major ovarian hormone is the estradiol, and it works to maintain various female reproductive structures, including the oocytes, the eggs that are found in the ovary. It's produced by maturing ovarian follicles, and it induces a surge of luteinizing hormone that's involved in ovulation. It also promotes the development of female secondary sexual characteristics during puberty. For male hormones, the most prominent male hormone, of course, is testosterone, which is produced, as you might guess, by the testes. It stimulates the development of the male reproductive system in fetuses and adolescents and promotes the sex drive and sustains sperm production. Inhibin limits follicle stimulating hormone secretion in order to regulate sperm production, so there isn't an excess of sperm being produced. So to review, the endocrine system is a system of glands that secretes hormones into the blood. Hormones bind the hormone receptors on cells and stimulate changes in the cells, which affect their activity. Hydrophobic and hydrophilic hormones have unique pathways for stimulating a target cell. Hydrophobic hormones can pass directly through the membrane, but need to bind to a receptor intracellularly and produce a hormone receptor complex, which is then able to enter the nucleus and directly influence transcription and translation. Hydrophilic hormones instead have the plasma membrane as a barrier. Therefore, they're going to bind a receptor that's found on the cell's surface that is G protein coupled that will activate adenylyl cyclase and increase cyclic AMP levels, and will in turn influence through phosphorylation of different proteins, the activity of many different proteins found inside the cytoplasm. And then finally, there are several major glands that regulate body functions through the production of dozens of important hormones, and that's what we just spent the last 20 or 30 slides reviewing. So this is the end of the first unit. Please review your notes, and then you should proceed to do the worksheet that will be linked in Blackboard.